Oh, we have to clear one last thing on you.
Oh,
You scroll back over to this window.
where's my mouse now? Is it? Oh, there it is.
There it is.
Okay.
And then this gap.
Got it.
Yep.
Thank you.
Yeah.
I'll be in all the runs.
I don't know where the microphones are. Is it acceptable?
Is it okay?
Just checking it. All one.
Hi, good to see you. I don't know how to get around here.
Yeah, I met I've actually seen about
I am.
Yeah. I love that. Oh, I have to. That's right. Yeah, true. It's been a while though. I have. Yeah, doing it. Yeah, I thought about it. It might be there. Yes, it might be. I would love to see this because I can ask the yeah, I can share a presentation too. It's pretty interesting. Yeah, I'd be happy to. Yeah, you're right. I'll make sure they get forward to it. Well, it's not great, I guess. Better to be sort of when you're doing it, thinking about it as opposed to just recording it. Yeah. Yeah. I know. And I like doing my options. Yeah. Well, I was trying to chat with you later, and then are you doing something? Yeah, I think so. Yeah, I think so. So lots of us think it's too crazy. I'm gonna take the ball. It's not. It seems like it's melting down. Yeah, they're totally fine. So it's a place to be in this. Yeah, the front range is better than yes. I don't think so. Maybe some students. I think I have one student has I don't know. It's been traffic so much. But I feel like I have stuff I need to do at home. Yeah, I know. It's but it's fun. It's totally fun. Totally good. Yeah. Yeah.
Well, welcome all. We're just a little bit after 1 p.m. today. It's my distinct honor and privilege Association for Environmental Engineering and Science Professors, distinguished lecturer for the 20. Professor Elizabeth Edwards from the University of Toronto. A little bit about Professor Edwards' background is that he received her initial degree in chemical engineering at Yale University, and then went on to get her PhD at Stanford University in civil environmental engineering. Personally, I got to know Professor Edwards for a lot of his work with organophosphate restoration, especially for the waste, and the work with, he ended up going with CP1, which became a commercialized product through serum. And it's always fun to work with anaerobes because they end up doing very interesting biology and chemistry and bio. And so today we'll actually have the opportunity to hear about that because part of the AESP lecture is where there's two distinct options that we can select. And we'll be hearing from one of the historical and long-term aspects of the research that's going in as Edwards Laboratory on the bioremediation within these anaerobic realm, where the other option was the anaerobic. So if you have the opportunity to speak with Professor Edwards, he has a lot of research on them, and there are fun labs here as well. And I will hand the floor over to you.
Perfect. Turn this on. Just testing. Can you hear me okay? Okay. So thanks, Preston, and thank you all for being here. And I'm yes, thrilled to tell you the story. I call it Adventures in Anaerobic Bioremediation because I've been doing this here since I was a graduate student, like many of you. So and there's been lots of fun both on the applied side and on the fundamental side. So I am at the University of Toronto. I'm in chemical engineering, cross-appointed in cell and systems biology. So I get some students with a biology background, which was kind of helpful. And some years ago, because the University of Toronto is huge, it's close to 100,000 students now. Yeah, it's huge on 3 campuses, and it's very medically oriented. There's a big med school, and there's a number of hospitals around. And so there was really, most of the microbiology on campus was medically oriented. So we formed this center called Biozone, really to kind of be a node or a clearinghouse for non-medical micro happening on campus, basically, because you could find people in earth sciences or in food engineering or somewhere like that. So that's the story behind Biozone a little bit. I might tell you some more in a minute. Before I get started, I definitely wanted to give a shout to the AESP for giving me this chance to go around and tour so many places that I've, met people from all these different schools at conferences and so on, but actually having gone to the universities and seen, met people there, it's been very special. And especially all the students and postdocs that I've met, it's been really fun. And AESP has this vision of being a collaborative, supportive, and diverse community of researchers and tackling, these challenges in water quality and air quality and remediation, so on, that we're all looking at. So it's totally up our alley. I joined when I was a grad student, when it was AE. There's no S. There was no science in it. It was just an Association of Environmental Engineering Professors. And then later on, it recognized that science was important. We need to understand mechanism. We need to understand. Need to make it broader. So I thought that was a good addition. Okay. So I really thank everybody for this opportunity. And the story starts from just understanding the way I got into it when I started my graduate studies was really, well, we know we have these spills in the environment and these pollutants migrate in various ways in the subsurface, but typically the groundwater becomes anoxic. And so just because you have a large amount of organic carbon there, and so you tend to consume all that oxygen, and then it doesn't re-aerate very quickly. So the question was, what is the fate of these contaminants under anoxic conditions, and particularly what is the role of biology? And so for the last 30 plus years, I've just been putting dirt in a bottle anaerobically. And it really is, just a lot of different dirt from different places. And it's been so much fun. And it's amazing what you can learn from all this. And so we go to contaminated sites. And when I finished my PhD, I went out and worked in consulting. So I had access to, colleagues who were working with clients who had contaminated sites. And so it's pretty easy to find anyway. And so if you look at this slide, these are the kinds of chemicals that we've looked at over the years. And in the two boxes here in blue and in red, so the ones that are in blue here are relatively reduced compounds, like these aromatic hydrocarbons. So they're good electron donors in the microbial metabolism. And the ones in red that are chlorinated or halogenated, they tend to be better, a bit more oxidized, they tend to be better electron acceptors. And so you kind of look at this way, and certainly I did my PhD with Perry McCarty, and so some of you probably think about microbial metabolism this way as well, but every cell needs something to eat and something to breathe is one way to think about it, or an electron donor, an electron acceptor. And so when you just want to think about microbial physiology in sort of a unified way, you can think of it as requiring, every organism requires an electron donor that's relatively reduced, that gets oxidized, and then those electrons get transferred to an electron acceptor, and that electron acceptor gets reduced. And so many substrates are good. I'm trying to figure out where this mouse goes. It only goes on this one? I'm just trying to figure it out. Sorry. Is it on both? Okay. Anyway, yeah, okay. It's just moving around a little faster than I can see. So lots of electron donors, we commonly understand sugars and so on, but microbes can do all kinds of interesting things, using inorganics as electron donors, using oxygen and aerobic respiration, but all these anaerobic electron acceptors that I'm sure you're very familiar with, nitrate, sulfate, iron, and CO2 reduction to methanogenesis, or even CO2 reduction to acetate. All these things are possible. If there's a free energy release, then microbes can grow. And that's the essential system that is a nice unifying concept for all this. And so as Preston mentioned, we studied a lot on dechlorination. And so if you look at this concept of organohalide respiration, it falls right within this realm, where the microbes that do this organohalide respiration or dechlorination reactions, for example, tend to use hydrogen as their electron donor, not only, but tend to then use these chlorinated solvents as electron acceptors in their metabolism. And so tetrachloroethene to tri to di to vinyl chloride, and vinyl chloride to ethene. And just so we put this on this same footing here, this dechlorination reaction actually releases quite a lot of energy. So one step in the dechlorination here from tetrachloroethene or PCE to TCE, trichloroethene, releases 192 kilojoules per mole. And it's negative. So, so cells have exploited that, and that's just one step. So imagine the 4 dechlorination steps. Okay? So it's quite a lot of energy. So that's that background that I wanted to provide you with. When I started to work on remediation, we were looking at hydrocarbons. And so let's look at this from the perspective of something like benzene as a model hydrocarbon, okay? And so hydrocarbons are really great electron donors for many microbial processes. And so you have here the electron accepting reactions here, oxygen, nitrate, iron, sulfate reduction, and methanogenesis, all with benzene. And you can see the delta Gs here are very high, 3,000 or more, roughly for these first 3 cases. And under methanogenic conditions, there's a lot less energy there, but the sign is still negative. Okay, so that means that you should be able to find microbes that can do this, even though the delta G is so low. Okay, so in theory, they're great donors. And in fact, from the bioremediation perspective, aerobic hydrocarbon bioremediation, which is this first gray line here, is the most widely used form of, industrially anyway, the most widely used form of bioremediation out there. You just take contaminated soil and make a pile and aerate it like a land farm situation or tilling situation to encourage aerobic microbes that degrade hydrocarbons. And so that's probably the widest use of bioremediation currently. I just put one little more line here for toluene, very related compound to benzene, just methylbenzene. And you can see that the free energy under methanogenic conditions here is very similar, okay? That'll come up in a little while why I'm mentioning this right now. So benzene has been known to degrade aerobically for a long time. In the probably in the early 50s, people even described aerobic benzene aromatic compound, aerobic degradation of aromatic compounds like benzene. And what you see here is some stoichiometry at the bottom. Not only do you need oxygen because these microbes are aerobes, so they need to breathe oxygen, they also need oxygen as a reactant in the activation of the benzene ring, okay, to form these catacols here. And if you write out a stoichiometric equation here, you can quickly realize that for 1 milligram of benzene, you need 1.5 milligrams of oxygen, and you produce over a milligram of cell biomass, okay? And so one of the challenges then for thinking or invoking, remediation in situ for hydrocarbons is that you have to get a lot of oxygen in there. And you also produce so much cells that you're likely going to clog everything as well. And that's what happens if you try to stimulate this too much, because you get so much biomass growth. And that's because there's so much energy available, okay? And so then the question becomes, well, what about anaerobic conditions? And because this reaction happened so quickly and was well understood, when I started my PhD, it was really the dogma of the day was that these molecules are completely recalcitrant under anaerobic conditions. So benzene, why do we have oil reserves? Why are there hydrocarbon reserves that have been there for millions of years? That these molecules are just not touched in the absence of oxygen. I think the first documented case where that got a little bit put on it, started to be questioned, would be in some studies, some bottle tests that were done in the wake of the Aramco Cadiz oil spill in the English Channel, and where C14 labeled benzene was used, and then some C14 labeled methane was detected in these bottles. So that might be the first case, but really the real first really vetted data was a study conducted by Tim Vogel, who was at Michigan, now he's in France. He was a student of Dunja Gabor-Galich. He was a professor at Stanford, my co-advisor actually. And they had found some enrichment cultures that at the time they were looking at lignocellulosic degradation. And they found some enrichment cultures on ferrolate that transiently accumulated toluene and benzene, interestingly. And they made these enrichments on benzene and toluene and were able to show transformation under methanogenic conditions. And so these were published, you can see the years, quite some time ago now. So it was in this backdrop that I came to Stanford, okay, with my little daughter in tow. She's 36 years old now, just to let you know. And I went to work with Perry and Dunja, and the question was, could we repeat finding anaerobic benzene degrading activity, because those enrichment cultures that they'd been working with were no longer active, and they wanted to try it again. And so I set up, so that's where I first started putting dirt in a bottle, okay, anaerobically. And so we were able to easily find methanogenic toluene and xylene degrading cultures. This happened, in multiple samples from many sites, and that was not a problem. It was much more difficult to find benzene degrading cultures. You had to wait a lot longer, but if you wait, you can be rewarded. And I happened to go on maternity leave, and so I was out of the lab for a while. Came back and sampled these bottles I set up, and lo and behold, the benzene had gone. So that was I was able then to continue to study this process. So sometimes it's good to take a break and wait. And so that's the origins of these cultures. And in these microbial cultures, what's actually going on in these methanogenic environments, as many of you know, the methanogens themselves really only use simple substrates like hydrogen and acetate and a few others, but primarily that. So anything that you are converting all the way to methane has to go through this food web, these trophic groups. And so same for benzene or toluene or xylene, some organism has to first ferment these, perhaps to some intermediates, or perhaps all the way to hydrogen and acetate, and then those then get consumed. And so we really are interested in this organism number one here. What is initiating the attack on these substrates? And the community this is a picture of the xylene degrading culture at the time. And so it is a complex community. And the reactions I'm going to show you right now that the reactions to convert benzene to hydrogen and acetate are really not favorable unless you remove those products. So it really is what is the definition of syntrophy. So I give this talk to many groups, and some of the groups have more biology than others. And so hopefully I'm not boring you too much. But you know, this concept of syntrophy to me is I mean, I have so many things in the lab called syntrophy because I just love the word, right? Computers and freezers and everything else. But it's just because it's just such a neat concept. So I want, and we tend to overuse the term perhaps in defining what a consortium is. But you know, here's the example for benzene. So organism number one is taking benzene, presumably to acetate and hydrogen. Okay. And then we have our methanogens, the acetoclastic methanogens here and the hydrogenotropic methanogens. And so those are their overall energy equations, that overall energy equation I was talking about. And so the free energy changes here, the big thing to note is that this, at standard conditions, this reaction is endergonic. There's no energy to be gained from this first step. The methanogens, there's no problem here, standard. And the overall is that minus 62 I showed you. And then if we reduce the concentration of hydrogen and you reduce the concentration of acetate in these systems, okay, you can find concentrations, and I'll put them here. This is standard conditions versus, 10 to the -4 atmospheres for hydrogen and a millimolar instead of a molar for acetate. Then you can find conditions where the free energy, that's still that minus 62, is evenly distributed between these three guilds. And that's actually what happens in these anaerobic environments, that they just naturally poise those intermediary concentrations such that this can happen. And that really is the definition of syntrophy. But it does mean that it's very difficult to isolate organism number one, because you have to find ways of really withdrawing the products in order to get that activity to happen. Otherwise, you won't see any of the growth. So syn together, troph is eat. So it really is dependent on the substrate. And the same thing happens for propionate and benzoate and some other compounds that have a positive free energy if you build up those products. Okay? So glucose is not like that. So that is where I took it at Stanford. Before I move on to the next story, I just want to take a moment to remember Dunya Gabrigalich. I said she was my PhD supervisor, but she passed away the year I graduated. Which was very sad, but she was the most amazing person. This is a building that doesn't exist at Stanford anymore. It's called it was the Terman Engineering Building in the pond. And every year ducks would have chicks in the pond, and she'd have to build a ramp so the ducks could get out of the pond. She had a hummingbird feeders in her window, and one time there was a hummingbird that had broken his beak, and so she cut the hummingbird feeder so he could feed still, and she gave them all names. Anyway, she was this kind of a person. And she also tended the rose garden in front of the windows around the campus and around the building. So if you go to Stanford, there's a plaque in her honor in the rose garden. And so she was truly she was a trained classical anaerobic microbiologist from Zagreb. And she really, I mean, I feel lucky to have been trained by her. And then Perry was my co-advisor. So I had an engineer and a microbiologist, which is a perfect situation. Okay, so when I finished at Stanford, I wanted to know whether this bioremediation thing even happened in real life, and whether industry really was making any use of it. So I went and worked in consulting. And when I started working in consulting back in Canada, the contaminants that were then the most prevalent and the most concerning were these chlorinated solvents. And so we worked first at to characterize, the extensive contamination at a lot of sites. But just before that, when I was still a graduate student, it was well known that dechlorination could happen. So these dechlorination reactions like this have been known since the 70s even, that if you had PCE in groundwater and there wasn't oxygen there, that you'd get these dechlorination reactions. Okay, so it did require anoxic conditions and then the problem with this was that it would produce things like vinyl chloride that are more toxic even than the parent compounds, okay? So the things that were spilled at the sites were PCE, which is dry cleaning solvent, and TCE, which is your industrial degreaser. For any time you have to weld metal parts together, they use TCE to clean it, so in automobile and semiconductor industry. So these are the things that are spilled, and these are the things that show up in the water. And it certainly made a bad situation worse. And I remember talking to consultants and they'd say, yeah, you just don't want to spill anything in an anaerobic aquifer because you're going to get these problems. But if it's aerobic, then you don't get so much of these dechlorination. But it was Jim Gossett and Dave Friedman at Cornell who really blew this field open when they, studying the Ithaca anaerobic digester, found enrichment, culture, or found a mixed culture that could dechlorinate vinyl chloride to ethene. And so this was a crazy, a huge change in the paradigm that actually maybe these dechlorination reactions could actually lead to detoxification. And I remember I was a graduate student still when this paper came out, and Perry McCarty ran into our lab meeting, holding this paper above his head, shaking it, going, this is really important. This is a remarkable discovery. And I know most of us were kind of, we didn't understand the implication of it, but Perry certainly did. So as I said, after working in consulting, one of the first things, I was working in consulting shortly after this. And one of the things we went to do was go out to field sites and just see if actually ethene was there. So this was shown to happen in an anaerobic digester, but does it happen in the field too? And so interestingly, when consultants would go out and take samples for groundwater and they send them to a lab to get analyzed, the only compounds they'll look for are chlorinated, toxic compounds and things like that. They never measure methane or oxygen or other things like that, or ethene. So we had to work up some procedures to get good measurements for ethene. But sure enough, when you go out to field sites, many of them you would find this. And so we went to one of these sites where literally it almost seemed like the ethene was bubbling out of the ground. Like it was really very evidently active, and put dirt in a bottle and added various electron donors. And before long, we had complete dechlorination of TCE here, all the way to ethene stoichiometrically. And we called this the KB1 culture and to me, I had been working on hydrocarbons anaerobically where things took months and months and months ago. And here was something that took a few days to a week to go. And it was quite surprising. And so my mom named it KB1 or kick butt because she had a pickup truck, a red pickup truck she called the kick butt truck. And because it was zippy. And so she said, well, why did you call it kick butt? And I said, oh, okay, KB. So that's how it came about. Okay. And so it's just these bottles, right? And you have a nice mass balance on here. You put in TC as electron acceptor. Methanol was what we used because it was a convenient co-solvent, also because I think the Cornell group was using methanol originally. And the products are ethene, the chlorine goes to HCl. You've got methane from the excess donor, and you get biomass. And so we've characterized that over the years. But, in the, what we wanted to know is, well, how prevalent is this ability for microbes to take TC all the way to ethene? And what we would find is that at many sites where we put dirt in a bottle, you wouldn't get complete dechlorination. So some sites you did, and some sites you didn't. And that was kind of confusing. And remember, this is quite some time ago when the sequencing of DNA and all that kind of stuff wasn't quite as readily, not nearly as readily available as it is now. But so, some sites, no matter how long you waited, you'd see partial transformation. TC would go to cis DCE. And we're adding all kinds of electron donors. We had methane producers. No, the microbial community was very active, but the cis DC was not going any further. So that's where we had the idea as well. We have some bottles that dechlorinate and some bottles that don't. Let's just mix them together. And so when we took some of that culture that we had enriched from the other site and mixed it in, right away, we had complete dechlorination to ethene. And so that was our first sort of clue that, there's this notion that microbes, everything is everywhere, and the environment selects. And this was a first clue where maybe that doesn't hold, or maybe we can't wait that long. Maybe it would eventually select, but it might be too much time. And so then, based on this and other data we had, my colleague Dave Major, who's at Geosyntec, the consulting company, he was my boss before I went back. Oh, in the meantime, I went back to academia. I forgot to mention that. But I did go back to academia at one point after a few years because I was desperate to put dirt in a bottle in a lab so I could test out these things. Anyway, so Dave Major was able to write some proposal to ESTCP to some funding to do a field demonstration of bioaugmentation at the Kelly Air Force Base. And so this was a real field experiment. We had one plot here, and we had two plots, one that we bioaugmented and one we didn't. And then this one here has extraction wells and an injection well, and we'd extract from the back and inject in the front, and then there were monitoring wells. And so it was maybe like, imagine a thing the size of this room with wells at one end and the other, and we extracted and recirculated and through this system, about roughly the size of this room, probably underground. And we injected 15 liters of the culture once, and then monitored. So there's the culture that we injected in a little beer keg like this, injecting it anaerobically. And so here's results from one of the monitoring wells where we had PCE transformation to cis-DCE before bioaugmentation, and that was true in the control plot as well. So that partial dechlorination was happening at the site. We knew this already. And then when we bioaugmented, we saw this complete conversion. So that was very exciting. And if you look at this paper here, it involves a whole bunch of people, but there's Sandra Dworczak. I can't read the names, but Sandra's on there. And anyway, so on the basis of this, Dave Major and Evan Cox and a number of others decided, hey, maybe there's a business here. Maybe there's a business in not only, doing this bioaugmentation, but also doing these treatability studies to be able to evaluate whether a site needed help or not. And so together we founded Serum, and Serum hired Phil Dennis, who was one of a student at UT that I co-supervised, and Sandra, who worked with me. And as I said, Dave Major used to be my boss when I was in consulting, and now we were, so it was this. Anyway, we started Serum, and at the beginning, it was kind of tough because a lot of people didn't believe in bioaugmentation. There'd been a lot of snake oil salesmen in the business before that, kind of selling bags of bugs that would have these miraculous properties. And so in the industry, people really didn't believe that you needed to do this. And so we spent a lot of time convincing people that there was a reason and a fundamental reason and a scientific reason for why this would work. And so what happened since those early days in these intervening years, not just around the world, that this notion of bioremediation, first of all, and also for chlorinated compounds, really blossomed, and a lot of research groups got on board with this. And there's just sort of what's been so much fun for me and for many others is that in parallel, we had experiments to look at the practical application. How do you actually grow more culture to be able to inject in these sites? What are the regulatory approvals you need? They vary from jurisdiction to jurisdiction. What do you need to do? How do you deliver this so you don't kill them as you're delivering them, exposing them to oxygen? What are the impacts of other co-contaminants that are at the site? All this kind of stuff. And then on the fundamental side, we're still trying to figure out what are these organisms? Where do they come from? What can we learn about their physiology and their enzymes and their communities and so on. And then at the same time, over these last intervening 25 years, there's been a genomics revolution. And so the tools by which we could interrogate these cultures have also improved. And so it's just, I just been on a roller coaster ride basically for the last 20 years. And it's been so fun. So here's a kind of a summary of what that has led to on a little bit on the fundamental side, but so here's for PCE anyway, the dechlorination pathway. And as I told you, 1989 was a key date from going to poison to fruit ripening hormone, which is what ethene is. And then the first organism that was isolated, so the microbiologists say, oh, well, this is a microbial process. Let's find those microbes. The first organism isolated was called Dihilobacter restrictus. And its only known substrates are hydrogen and PCE and TCE. And the only thing, so that's why it was called restrictus. But it also only took PCE as far as cis-DCE and didn't go any further, okay? And so stopping here. And then a whole host of other microbes are only doing these first two steps in the dechlorination. And so it turns out that the realm of doing these last detoxification steps really happens to be in these organisms in the genus now called Dihilococcoides. It was first isolated by Steve Zinder and group at Cornell. like 8 years later after they'd actually had the enrichment cultures. It took that long. I remember Steve said to me, oh, you know, you have an isolate when it doesn't grow anymore. And so it's really tricky to get there, but he got there. And then as late as 2015, Frank Loeffler's group has shown that some members of a related group of organisms called the Dihalogenimonas can also do this vinyl chloride caffeine reaction. And so we're still learning, but you know, these are tricky organisms to grow in isolation, but they grow very well in mixed culture, is the bottom line. Of course, then genome sequencing at the same time. And so the first genome of Dihalococoides ethanogenes, now McCartyi, it has been renamed, was sequenced in 2005. And it's tiny, a small genome, like a third the size of, say, E. coli or something like that. It's highly specialized for these enzymes or these genes here that code for enzymes called reductive dehalogenases. Within the genome, there can be anywhere from, well, in this genome, there was, I think, 17. And then the other part is that it was known also through some biochemistry that was done painstakingly on these organisms early on, that the enzyme requires vitamin B12 as a cofactor, yet the organism doesn't make the vitamin B12. There's no gene, and it's a very complex pathway and very easy to recognize, and it doesn't make it. So they must scavenge B12 from the environment. So all these things are helping us to understand when we apply this in the field, why or why not things might be happening. So these reductive dehalogenases are a very interesting group of enzymes, and they're all different. It's not like it's a duplication of them here. They're all different. And so the notion is, well, I wonder what other substrates these organisms could actually tackle. And then through collaborative work from around the world, you know, it's starting to become more clear that there are different organisms specialized for different substrates. So this is a slide that originally was started by Frank Loeffler, and we've kind of worked on it together to increase all the things we know. But the chlorinated ethenes are here. But there's chlorinated ethanes and chlorinated methanes, chlorinated propanes, chlorinated benzenes, lindane, PCBs, and this is now already out of date. But what all the research has found is in gray are the organisms, and in green are the reductive dehalogenase genes. And so now you can go out to a site and survey for the presence of these genes that are required for these different dechlorination steps. And that's been useful for it's really cool to go out and speak to the site engineers, you know, and they're quite interested in understanding about, you know, is my site positive for vinyl chloride reductase, for example, you know. So that's been a lot of fun. The other thing that's interesting, though, is that, you know, you'll have Dihalococoides and Dihalobacter and these organisms that are now, we appreciate them as obligate organohalide respiring bacteria, meaning the only metabolism they can do is this dechlorination. The only electron acceptors they can use, which also has implication for why bioaugmentation can be successful. And then no two are the same. So one Dihalobacter would be very different from another because they have a different complement of these enzymes. And same for Dihalococoides, Dihalogenimonas, and many others. Okay, so here's a picture of the serum labs as of just very recently, actually. And so this is how they grow the cultures. So remember, you're growing anaerobic cultures on volatile organic compounds. And so you need to make sure that everything is sealed at all times. And so they use these large beer kegs. These hold 100 liters. And then they also have now they have a 1,000 liter and a 2,000 liter reactors as well. And this is Sandra Dwortzek here and Jen Webb. Jen Webb is really the mother of all these cultures. And my students were there on a visit. And so as of last fall, the cultures have been deployed at more than 850 sites around the world. So this is kind of amazing and to think about. The chlorinated solvent, the KB1 culture, and they call them the KB1 plus suite because we also have cultures that we've also enriched on chlorinated ethanes and chloroform and other products. Okay. So Olivia was a PhD student in the lab and was very interested in closing genomes from metagenomes, like some of you I know have spoken to have done. And so what I'm showing here is just a graph with time and amplicon sequences, meaning phylogeny. So the relative abundance of different organisms in the culture. In green are the dihelicocoides, and in blue are some methanogens. What you can see is at some point in time, this is time when on this axis, at some point in time, there was a switch from one dihelicocoides to another. And so I'm just showing that to say that, when you take an enrichment culture versus an isolate, you have this functional redundancy. You have many different organisms that doing ostensibly the same thing. And we were totally surprised. We knew we had at least a couple of strains of dihelicocoides in the culture when we first were doing old-fashioned clone libraries and qPCR. But when we started doing actual metagenome sequencing, Olivia was able to close 8 different genomes of dihelicocoides, and there were still more pieces that weren't closed. So at least 8 different genomes that belonged to different clades. So there was Cornell and Pinellas, for those of you who know what that means. But there was different clades, and all of them had different complement of reductive dehalogenases in their genomes. But what we saw was that the genome organization was identical. So in other words, the order of the genes in the order of the reductive dehalogenases in all those genomes. It wasn't, we had originally thought that it was just like each strain picked up a random complement of these different dehalogenases, but they're actually very synchronous, meaning they're all in the same order. So that tells you something about the evolution of these organisms. And so, the take-home message is that learning this very fundamental architecture or building, construction or makeup of the culture helped us understand why it might work well in the field. Because when you inject a culture into the field, the conditions are always different at every field site. Sometimes the pHs are different, or the different co-contaminants and so on. But if you have many, many strains there, maybe one of them will be able to survive under those conditions and still grow. So I think that's part of it. And that's true also for the cetogens and methanogens in the culture as well. So that's one message that I wanted to share. And we also created a database of all these RDases. So there's these reductive dehalogenases to help organize them, because people would always call their reductive dehalogenases number 1, number 2, number 3, based on which ones they would find in the genome, as opposed to what are they most similar to from others. So that helped organize that data. So these reductive dehalogenases are fascinating enzymes. This is the abbreviation is RDH for reductive dehalogenase homologous enzyme. And there's a subunit A and a subunit B, which is an anchor protein, holds it into the membrane. So this is the cytoplasmic membrane. They sit facing outside into the periplasm outside of the cell. And so this is outside and inside, and somehow the electrons from hydrogen through a hydrogenase get transferred to the reductive dehalogenase. There's been a lot of work in that area, but I'm not from others. I won't get into it right now. But there are thousands of putative sequences. If you do metagenome sequencing, you see many, many, many sequences that are homologous to these dechlorinating. So there's many out there. So we'd love to mine them. We'd like to know how do we find out what their functions is? We know about a few. We know vinyl chloride and PCE and a few others, chlorobenzene and chlorephanes. So how would we find this? What would you do, right? And so one way that people like to do, molecular biologists like to do, is you clone the sequence into another host and express the protein, and then you can test it, right? Because in the native host, so you take dihelicocoides, it has 17 different dehalogenases, but when you grow it on PCE, it really only makes 1 or 2 of those. And how would you induce it to make any of the other ones? We couldn't figure that out. And so the ideal way is to take the sequence now with all the modern tools we have now, you take that sequence of that reductive dehalogenase, put it into another host, make that host make it, and then assay it. So for that to happen, you need 2 things. You need an assay. First of all, you need to be able to test it. And then you also need to have the expression of a functional protein. And people had tried for a long time to express these proteins in E. coli, and it failed a lot. And partly it's because they're very oxygen sensitive. Partly it's because they have this vitamin B12 requirement. Partly it's because they have 2 iron sulfur clusters, and so they need that. And so it has been a long struggle. I had a PhD student spend a year trying to do this, trying all kinds of different tricks of biochemistry to get this. It failed, and I had to move him on to another project. But recently I had a student I have a student, she's still my PhD student, Catherine, who got an idea from a research group that was also studying chlorinoid or B12 dependent enzymes, where what they did is you co-express the vitamin B12 uptake pathway. So what that means, this BTU means B12 uptake, BTU. And so E. coli can't make its own B12 either. And so it has to pull it in from the environment as well. And it has a pathway to bring this giant molecule into the cell. And so it has a native pathway. But if you're expressing also this reductive dehalogenase and you're making it in high abundance, then you need a lot of B12 because you need one B12 for every protein. So you need more than what this this pathway could incorporate. And so this B12 uptake pathway, so this is vitamin B12, it's a cobalt-centered sort of a bridged porphyrin ring, largest biological cofactor there is, bringing it in through the outer membrane and the cytoplasmic membrane into the cell and into the reductive dehalogenase. As the reductive dehalogenase is being synthesized, it has to fold around this. Anyway, all to say, I wouldn't be telling you this if it hadn't worked, right? So, that's what Catherine did, is she expressed this enzyme, which is a chloroform reductase, in E. coli with the BTU uptake pathway. And then we have to assay it anaerobically using methyl viologen as an artificial electron donor. And then you look for the dechlorination products. And in doing that, so this is the experimental data here. This is chloroform was the substrate and dichloromethane is the product. So this is dichloromethane produced. And the only two conditions where she got dichloromethane produced was when you run the reaction anaerobically, you include the BTU pathway. And if you exclude the BTU pathway or run it aerobically, A is aerobic, okay? Or if you don't have the BTU pathway, then you don't get any expression. And then the other part of this delta iskra or lobster are two different strains of E. coli used as the host that are supposedly good for expressing anaerobic enzymes. The delta iskra doesn't degrade iron sulfur clusters and so on. So that's what they are. They're just molecular biology tools E. coli made to help. Anyway, so it works. So this is great because now we can take sequence information from our metagenomes and plug it into E. coli, make a lot of the protein, and then start testing substrates somehow or another, okay? And so that is great. There's one caveat though. We've been able to do this, and Catherine is doing all kinds of work on this right now, testing on lots of different reductive dehalogenases, specifically from Dihilobacter. When we try to express the Dihilococcoides RDases, they're not working. And so for whatever reason, there's something else that we need to tweak. So I have a new PhD student who's going to work on that. But it has offered incredible insight. We can already make mutational studies. So we can make, you can mutate one amino acid and see how much it affects the activity. So that's been, that's in progress. So this is this TC dechlorinated culture. And so you can see, I like to call this a tale of two communities because I told you about benzene before. And now when we're looking at KB1, what's actually going on is we provide some fermentable donor. And in the field, when they do the remediation, it's any donor that's cheaply available, really, that they use, gets fermented to the hydrogen needed by Dihilococcoides for chlorinated solvent dechlorination. The methanogens and the acetogens provide that vitamin B12. The acetate is a carbon source because they need a carbon source. So this is a lovely community. And then in the benzene case here, here we have benzene as our pollutant and we're making it. But the parallels in the community structures are as you can see. And so I want to just go back for a minute to benzene because I've been working on that since I was a graduate student. And there's still some big puzzles left to solve, and maybe you can help me. Way back in 2003, my first PhD student, Anya, she did an old-fashioned clone library and from a mixed benzene degrading culture, methanogenic culture, and found one clone here that she called ORM2 for oil refinery clone, methanogenic clone number 2. And she said that would be the organism. I think that's the organism that's initiating, that organism number 1 that's initiating the attack on benzene. But the culture overall had a very slow doubling time and so on. And so it's hard work. We had small bottles and hard work. And this was a slide directly from a presentation I gave way back when at ISME in 2004. where I just plotting number of papers published that report anaerobic benzene degradation versus anaerobic toluene degradation, for example. And what you see is at some point here, I put these stars, there was an inflection a little bit where a lot of more papers began to talk about toluene degradation, anaerobic toluene degradation. And why, and the graph doesn't go on, but it goes way, way up. And the reason is because the mechanism was understood finally. And so in these years, it became this reaction now that we call fumarate addition became well known. And it's a really interesting reaction. So the methyl group of toluene here adds across the double bond of fumarate to form benzyl succinate. And so this fumarate addition reaction was described first in 96, but then subsequently has been found to be applicable for any compound with a methyl group, whether it's an alkane or an aromatic, substituted aromatic. And so really unblocked or completely demystified why, or confirmed that these things could be transformed anaerobically and are oxidized this way with a metabolic intermediate. And so, but you can't invoke this for benzene because there's no methyl group. And so what is going on with benzene? And so this is the puzzle that I'm still working on at this time. We have cultures that take benzene all the way to CO2 and methane, and we still haven't got a clue how they do it. Okay? We have a few clues, not that we don't have any clues, but we have a few clues, but not a smoking gun. So, when I went to Germany with some of the labs that are really good in anaerobic biochemistry, and they said to me, well, Elizabeth, yeah, this is great, but you don't have enough biomass. We're not going to be able to do any biochemistry until you have more biomass. And so I said, okay, well, we have 200 mils. No, that's not going to do it. And and it takes 30 days to make another 200 mils, or maybe more. It takes actually months because it takes a long time to grow. You can only do small transfers. So way back in 2011, I wrote or maybe a little bit later after 2011, I wrote a proposal saying, look, I spoke to Serum, my friends at Serum, and I said, do you think we could use your reactors, your 100-liter reactors, or try and scale up with some of your equipment? And they said, okay. And then I wrote a proposal, and so and got funded, luckily. So then we've started the scale-up process. So in 2011, we had about 1 liter of culture, and by 2016, we got our first 100-liter. And once you have 100 liter, it's a whole lot easier to make another 100 liter. It's made exponential growth is always, every time I see it again, it surprises me again. But and so now we have many hundreds of liters of culture. And at the same time, something else happened, which was as we scaled up, the rate of benzene degradation increased. So as we got to these larger vessels, fortuitously, the rate of benzene degradation is still not fast. Like if you look at this scale, this is months, okay? Just telling you, okay? And so we're just refeeding benzene over and over and over and producing methane. But as this was happening, we suddenly got the idea, gee, at the same time, I had a colleague in industry working for an oil company who talked to me at a conference or something and said, we still have benzene plumes. Even though benzene goes aerobically really well, we have high clay, we have there's still a lot of benzene hanging around. And I said, we have this culture, would you be interested in maybe testing it out? And they said, yeah. So we got some more funding then to do some field trials of seeing whether these benzene cultures could help kickstart benzene remediation in the field anaerobically, where you can't get oxygen in. So that when we scaled this up, we decided to call the cultures DGG in honor of Dunia Gabrigalich. So we call them these DGGB cultures. And I also, from when I was a PhD student, still had the toluene and the xylene culture that you have. We still had those. And so I didn't think, toluene in the field, it goes relatively well. So benzene tends to be the one that sticks around, but toluene degrades relatively well. So we never thought there'd be a use for the toluene culture in remediation, or the xylene cultures, but we scaled them up anyway. So we've scaled all of them up. And it turns out that when you mix them all together and inject, they work better together than by themselves because you need toluene to go first before the benzene will go. So it turns out, and same with the xylene. So it turns out that all of this has worked out really well. We've characterized. So Courtney is a postdoc in my lab right now, a super postdoc. really. She's applying for faculty positions now. And we've done the metagenomics of all of these cultures. So we have this toluene enrichment culture that I've had since I was a grad student, this O-xylene culture that we've had since I was a grad student, this benzene culture that I had since I was an initial professor. And we've done all the omics you could possibly think of at these, and now we can clearly identify the organisms. So there is a desulfosporosinus here that has a benzyl succinate synthase gene. And in the xylene culture, okay, there is a peptococcaceae that has a methylbenzyl succinate synthase gene. So they're very different. And the O-xylene culture doesn't degrade toluene, and the toluene culture doesn't degrade O-xylene, and none of them degrade benzene. Okay, so this is this kind of thing. And in the benzene degrading culture, there's a fermenting organism, that ORM2 that I mentioned before, that clone. She was right back in 2003. We're thinking of naming it Benzenivorax because that's all it eats. This culture, we can't feed anything else to it. It does not degrade, does not transform anything else, not even benzoate. Okay, the central intermediate to all this is benzoyl-CoA. And benzoyl-CoA is transformed subsequently all the way to methane and CO2, but all the way through to fatty acids here to acetate. That pathway is very well known, the downstream pathway. There are isolates that degrade benzoate. So there's that part of the pathway is well known, and all the genes and proteins are all expressed in these cultures for these downstream pathways. And then we can see these for the upstream for the xylene and the toluene, but for benzene, we see some very highly expressed proteins, but we have no clue how they're doing this. We don't know what the biochemical reactions are, but we know it goes from benzene to benzoyl-CoA without going through benzoate because the culture does not degrade benzoate. So it's kind of interesting. Okay, and then in the culture, there's also the methanogens, of course. There's a few syntrophs as well. And there's also a number of organisms that are involved in recycling biomass, amino acid, proteins, and others that belong to some of these candidate phyla, which are very interesting as well. And we see they contribute quite a lot in releasing nutrients back to the fermenting organism or the syntrophs. Okay, so we have these cultures. I've given them to Serum to do whatever they want with them. And so they have named them, as you can see, and scaled them up and put them on a nice diet, regular diet, and they are growing well. And so we've gone out to the field with them. And so the first bioaugmentation we did was in Saskatchewan at a gas station, a former gas station. And here the culture was injected at this top end here. So the groundwater is flowing this way. This is an old decommissioned gas station. And we also injected autoclaved culture down here as a control, and then culture up here and sampled. Okay, so this site had undergone a number of things. It still had quite a lot of contamination in it though. So that's what we did. And this is what it looked like at the time of injection. So it was winter, it was November, and snow on the ground. And here's the culture. And then you use a gas cylinder to pressurize the culture and push it down the well, like that, okay? And so here is some data from the site. This is an old gas station, I said, and they only go out once a year to monitor. So that's one of the challenges. But we have, these are the wells that were near the autoclave culture injection. And so what you have here is normalized concentrations because each well has slightly different concentrations of benzene. And so, you know, there's fluctuations, but not much. But at the wells near where we injected the culture, you know, there's a lot of fluctuations as per se, but overall, this is a log scale here. And so many of the wells are showing concentrations 1 or 2 logs below what they were when we started. So that's encouraging, but it was frustrating too, because they only would go out once a year, even no matter how often we pleaded. And so we decided to go to a plan B as well. And so we went to Canadian Force-Based Boarding. So those of maybe some of you, I don't know, but if you've been in the field of hydrogeology, the Canadian Force-Based Boarding is about an hour north of Toronto. And it's the site where there's been so much field work done to help define the field of hydrogeology, basically, and contaminant hydrogeology. So John Cherry, the book Freeze and Cherry, which is kind of the hydrogeology textbook from the 70s anyway, And the reason it was that is it's a nice sandy aquifer, but also you were allowed to put contaminants in. So you could put hydrocarbons, you could put gasoline, you could put PCE. So you'd have a known amount of mass that you'd put in the site, and then you could see if you could actually clean it up and how much you were getting. Because one of the challenges with real contaminated site is you never know the history and how much was actually spilled there. And then they have these sheet piling along the side here. And then sort of a collection well, so you could kind of contain what you're doing in the sheet piling, and then they're just using the natural gradient. So what we've set up is an experiment where between these two sheet piling, we have the natural groundwater flows in this direction. We actually have these diffusers here, which are tubes of piping that we put a NAPL, we put a mixture of toluene, benzene, methylbenzene, xylene, flow it through, and it diffuses into the aquifer. So we're creating a source, basically. And so that's what these cigar-shaped things are, the source that we're creating. And then we're going to bio-augment, or we have bio-augmented, and these rows here are multi-level wells, so we can get a whole transect. And then the bio-augmentation zones here, where we added on one lane here, we added the toluene culture only. And on the other lane, we added all of them, benzene, toluene, and xylene. So that way we have a control. We want to see that the benzene goes only where we added the benzene culture. Basically, that's the kind of experimental design we set up. So this is the site in Ontario. This is the day that this big geoprobe rig arrived and had to drive over these sheet piling here. This is the sheet piling back here. And it lifts up like that to be vertical. And then this is when the day we were injecting culture. But anyway, we got it to work. It was kind of crazy to driving this thing is self-driven in. I use a remote control to drive the geoprobe in and set it up. And then, so what's happening here is this is the drill rig, and it's the probe is going down here. So they push the probe down to a certain depth, and this thing is about 7 meters down. And then lift it up a little bit. And then Brooke here, who works with Serum, is putting culture into this little device they've built. So under pressure, so pressurizes the culture anaerobically into here. We were putting a ½ liter of culture at 4 different lifts or 5 different lifts of depth. We kind of spread it out over the site. And once she fills the little tube, using these quick connects, she takes it over to the well, the drill rig, and
water.
There's a T here, and so there's the T. So the culture goes into the T and into the well and down to the ground. Okay, and so then using gas, you're pushing it in, down, and then once we push the culture down to that 7 meter depth into groundwater, the surface. This is what she's doing now. She disconnects that, and then we chase that with anaerobic water to push the culture even further. Now we started doing anaerobic by adding production to that, and then just measuring 30 liters based on eyeball of how far down it goes, and that's basically. So we did that, and then now he's going to lift the well up a little, lift the probe up a little bit, and then that's what we did. So it's kind of fun out there. Unfortunately, this was done last August, so we're just still collecting data right now. And so we go out now every 3 to 4 weeks, collect samples, not only for concentration but also for DNA, and we're able to see if our but that'll have to be for another time. So in conclusion, then I just want to say that why does it work? Why do these organohalide respiring microbes work? Is because of this niche specialization. As I said, they're obligate organohalide respirers. The only thing they'll do is dechlorinate. So if you inject them in the ground, I mean, the worst case scenario is they're dechlorinating something that's not your primary pollutant, but at least they'll be dehalogenating something. Okay. Whilst other options, when you, in the past, when you're looking at aerobic hydrocarbon degradation, for example, the microbes that degrade hydrocarbons aerobically might be able to do a whole lot of other things as well. So it's not necessarily so specific. But each dihalococoides or each of these organisms are not created equal. They have these multiple dehalogenase genes, as I told you about, conferring some unique substrate range to each of the organisms. And there's this huge unexplored diversity that we need to tap into. And then, of course, for these strictly anaerobic BTEX fermenters or syntrophs, I should call them, it also might be a successful technology because these organisms are also niche specialists. So I was, I think I was thinking that they may, it's kind of hard to imagine that something can do benzene but not toluene, or toluene and not xylene, right? But that the specificity is like that. They have these unique hydrocarbon degrading genes and also unexplored pathways, as I told you. And so I think these are fascinating systems and extend, and maybe are simplistic to some extent because they're looking at single substrates, but might extend to a better understanding of some of our more complex systems as well. I also just want to spend a minute to just talk about the perspective of working with serum and geosyntac over the last 20 years. It's been so much fun, as you probably gathered, and we had these real problems like what's happening with vinyl chloride in the field? Where is this going? What we have this activity and trying to figure out why. So without meaning to, these collaborations were always very open. I shared the cultures with them with no strings attached, and we published the data openly. I've always published good or bad data about these things. It was very very easy to publish that way. And for serum, the advantage of having kind of academically vetted data was it actually turned out to be a really good marketing tool as well. And so it was a win-win, but we didn't mean it that way. And honestly, I never thought it would ever, the notion of that wasn't what we got into, was to make a company. It just became obvious at the time. And so over the years with this experience, and also my experience working with other industries, mining industry, pulp and paper industry, remediation industry, that we started to really explore this concept of open science. And so what does open science mean to you? You might think of it as meaning open access journals, right? That's one of the things, or open access source or code, computer code, and data and so on. Open innovation is when you have a consortium of companies that work together and share the IP or put the IP together. But at the very top here, it might mean, or it does mean, to just do your work and put it in the public domain without seeking a patent, without seeking to patent it, without seeking that that kind of protection on your idea. And so Biozone, I told you, was the center we set up probably around 2007. And all the labs are shared just like you guys and all that kind of stuff. But moreover, we kind of deliberately enter into our collaborations with companies by saying, look, this is the way we're going to work. You don't get any IP out of this, but neither do we. We put it all in the public domain and we can all use it. And the companies love it. Why? Because they get freedom to operate. They don't have to pay any license fee or anything like that. But that also means that I can continue to work on the same thing. I can, so it really means that you can continue to work on that, and they can continue to do their own thing like Serum has with what they have, but it doesn't impede. And really what companies are looking for is freedom to operate, freedom to use the information that they get, and then they don't have to share anything they don't want to share. And so that's been working really well. And during COVID, we started thinking about this a lot more because we have really tough problems we need to solve fast, all right, in this climate change world. And so the problem with patents and the problem with all that is that it really slows down the pace at which something gets transferred into industry. And so we asked the question, do you think this could help with some of these hardest technical problems that we need to get many, many people working together to try and translate this technology, a good technology, something that works. And we need to vet it. It needs to be reproduced in many, many, many times to gain that confidence. And these solutions need to be global as well, and so on. So we asked the question, is the current accepted framework for research at our universities, I'm talking about university-based research, is it adequately structured to meet these challenges? And so we answered some of these questions in this little paper. So if you're interested in this, we wrote this tiny little paper. We actually had 2 law students. So Kirsten and Sung were 2 law students who we hired to go over some of our agreements and terms and to do some polling of our collaborators, because we have about 50 partners, 50 industry partners in Biozone, to see why they partner with universities and what are they looking for. And the first thing, they're access to talent, new ideas, what doesn't work. They want to know, when you're working together, what is it that's not working? All the fancy science and nature papers, all the best data, what do you want to know? Hey, what is the real story behind all this? And then, IP and that kind of thing is much, much lower down their list of what they're wanting, why they would partner with an academic. And then as a group, when you're working together as students and colleagues, if you can chat about everything you do at conferences and everywhere else, it's a whole lot more comfortable, as opposed to having to keep a secret until you file a patent. So anyway, I'm happy to talk more about that because it's become a bit of a passion. Final slide, or almost final slide. I wanted to just tribute to Perry McCarty, who's the most outstanding individual and passed away just last year. And he was such a humble person, but probably the best environment. The way Bruce Rittman, one of his first students, says is he's the best environmental engineer that we've had in a long time around. And so certainly he had a knack of figuring out what the critical question was to ask about any problem and how to design an experiment for that. And just to illustrate that, as early as 1964, he wrote a 4-part treatise on anaerobic waste treatment fundamentals here with microbiology. And it still holds today. Like all the things that he said are still completely correct today. And then as early as 1967, he was looking at dechlorination of chlorinated hydrocarbons, or chlorinated pesticides, I should say, and really understood this reductive dechlorination process, and it was microbially mediated. And then as late as 2021, he's touting the values of secondary anaerobic treatment for energy sustainable wastewater treatment. And so truly a remarkable career. And so I feel you should look them up if you don't know. And so finally, I'd like to acknowledge all the many students that I've had over the years, and many collaborators, particularly my colleagues at Geosyntec and Serum, whom are friends, and funding from the US through the Strategic Environmental Research Defense Program, through the Joint Genome Institute, and then funding from our Canadian organizations, and NSERC is like NSF, the province of Ontario, and then an organization called Genome Canada that funds genomics. So thank you. There's lots of good books out there, and I'm happy to have questions. Yeah.
I'm really interested in
Yeah.
my question is, there's some fundamental reasons unfavorable and favorable effects of microorganisms, which is a feel that is a single microorganism have all of them.
So, I mean, you could say the same thing for any methanogenic system, right? Why have methanogens not evolved to use more complex substrates? And that's a question that's actually been asked, quite a bit. I think the reason is because if you can funnel everything to acid and hydrogen, then you only need to do that, and anything can come that way, right? So you have a strategic advantage. If you use one substrate, then you're wed to that substrate, or if you have to onboard that machinery. So it's basically like having an assembly line, and somebody is very good at what they do, and they can be very good at what they do, and not have to be very good at the other things. That's one answer. There is a paper from 1988 in the Canadian Journal of Microbiology, written by Mike McInerney, that's called Non-Equilibrium Thermodynamics, something. I can't remember, but the non-equilibrium thermodynamics in there. But what arguing too is that the fact of transporting in and out of a membrane, you can get energy too. And so by exporting acetate, for example, you can get a proton motive force or something like that. So there might be some benefit to using, exporting charged molecules or whatever, and that might you might get a little bit more energy than you would if you had it all within yourself. That's nothing. And also, you know, basically, I think of microbial cells in the community almost like organelles in a complex organism, right? And so by compartmentalizing, you make those reactions faster. Yeah. Yeah, but what would you do? So isn't enrichments and transfers directed evolution in some ways? I mean, we've been growing them for a long time. I keep trying to, you have no idea how many master students have just said, try this, let's put a bit of this in, let's put a bit of that in, let's put a bit, I mean, so many, you know, oh, I think it needs vanadate. Okay, let's try vanadate, plus minus vanadate. Let's try all these things, you know, to see, and none of them has dramatically changed the rates to try and make it go faster. It's gone a little faster, but the most dramatic thing was what I showed, which is scaling it up, which suggests to me that there's an edge effect and there's the headspace above, the glass bottle, things like that. There's some interactions there that maybe slow it down a little bit. But it's essentially enrichment cultures is a directed evolution in some ways. Yeah, you have to decide what selective pressures you're putting on. I would say yeah, well, benzene's maybe immune in itself. Yeah, you're totally right, and it could be. Yeah, I think it'd be kind of interesting actually to shine some light on it, or some UV on it, and see if that would speed things up, or yeah, maybe. The rate of we've done it for the KB1 culture where we sequenced 10 years apart the community to see how much it had changed, and the rate of mutations was not really that much faster than what would you expect naturally. So it didn't look like there was a lot of sort of enhanced mutation rates or anything like that. But it's a good idea. Yeah. The reality is the rate isn't really a problem because groundwater moves slowly, and what's a problem is consistency. So if it's always does it at the same rate, and I can predict that, and I make a transfer, and so this methanogenic culture is like that. We have another benzene degrading culture that does it under nitrate reducing conditions, and you can transfer, and half the time it'll go and half the time it won't go, and that's very, very challenging to figuring that out. So in some other cultures are a bit more ephemeral like that, and but these cultures have become very steady. So I think the rate. We're trying to understand why the rate is so low. I think it's because of those recycle. It looks to me like there's quite a lot of actually endogenous respiration or degradation that's happening. And I have a feeling that this benzene activation reaction mechanism produces toxic intermediates that kill the cells at the same time, or a fraction of the cells. And so you get a much lower yield than you would predict. And then there's a lot of recyclers in the cultures that I think you're getting. So that's what I suspect. And it makes sense. I talked to my chemist friends about taking a benzene and pulling a hydrogen off, which is what you have to do, de-aromatize this thing as well. And they say, well, it's not possible to do that at room temperature in aqueous solution. It's just not possible. And so the activation energy is too high. And so there is so whatever the enzymes are, and we have an operon with highly expressed proteins that we are pretty sure is involved, but we haven't been able to get an assay to work out in vitro to test this. Yeah. So for the PFAS question, I think that there is evidence that some PFAS can have some transformation, but as a remediation option, I find it challenging because there's too many compounds. For the reasons I explained, which is that one microbe does one compound, let's say roughly. So if you have 100 or 1,000 PFAS. I don't know how that's going to happen in the near future anyway, but it's still worth studying. What trendy compounds? There are lots of compounds out there, actually. There are antibiotics, there are, I think this notion, one notion that's come up is what is co-metabolic transformation? So what is co-metabolism? And Kristen, you might relate to this. You know, these very low concentrations of things, are they really co-metabolically transformed, or are they actually metabolically transformed even at these low concentrations? And I think that that's an interesting question, because there's this paradigm that we need to have a close substrate to get those things degraded, and maybe that's not true. So that's one question, I think, that maybe organisms can degrade at very low concentrations and turn around very slowly. Back to your question about rate, many microorganisms divide very slowly inherently, and there's lots of it, and you get this niche, and they just turn over once a month or once every 6 months and really happy and get rid of small concentrations of things. So I think that's the question for this. But any compound is interesting. If you have to have activity, follow activity. That's what I would say. Have activity first and then look at the microbe, not the other.
Those who are considered plants that are visually played. Could also give a solver.
Oh, I'm fine now. If you have.
Thank you all on that.